Bonjour tout le monde. We will start. It's 1:30. I want to welcome everyone to our session this afternoon. My name is Darlene Boileau. I'm on the board of the National Association of Federal Retirees, Ottawa branch. I want to welcome all of you, a very large number of you today to this afternoon session with us. I'd like to begin by acknowledging that the land on which the National Association of Federal Retirees, the Ottawa branch, office is located is traditional and ceded territory of the Algonquin Anishinaabe people. On behalf of the Ottawa branch, I'm pleased to welcome you to today's webinar, which is presented today by the University of Ottawa and entitled Concerned About Memory Loss, See if you're among the worried well. Veuillez noter que le webinaire sera présenté en anglais, mais la participation bilingue est toujours encouragée. Alors, on vous encourage de noter vos questions en français et on s'assure de répondre à vos questions dans la langue de votre choix. I want to give a special welcome to anybody here who is not yet a member of the association. We hope these webinars will inspire you to join us. And to our members, we hope that these webinars will encourage you to maintain your membership. I also want to remind our members about the annual mega recruitment drive, which is currently underway. Until December 31st, every time you successfully recruit a new member, you will receive a ballot to win one of 14 great prizes, including the grand prize of $10,000 cash, which is courtesy of Johnson Insurance, one of our great partners. All members who successfully recruit a new member will also receive a $5 gift card from Tim Hortons. To learn more, we would encourage you to visit our association website, and the link will be posted in our chat box for more information. There will be a question and answer period at the end of the webinar, and we would encourage you to type your questions in the chat box throughout the session, and I will read them out loud to the presenters at the end of the presentation. Also, we are recording today's webinar, and after it is over, Erin Higgins from our Ottawa branch will send you an email with instruction on how to assess the recording and the presentation slides, as well as all the contact links for the presenters and for the future studies that they will be engaged in. So all of the details and their information will be in that email tomorrow that you'll be getting. She'll also be sending you information on the mega recruitment drive and how to join the association or give the gift of membership. Now I'm going to turn the presentation over to our speakers. And I must tell you, I'm very excited because as an alumni of Ottawa University, I'm super excited of having Dr. Fraser and Talia Salzman with us today. And I'm very proud of the connection and the collaboration with the University of Ottawa that we are sharing this afternoon on this session. So today we welcome Dr. Sarah Fraser, who you'll see in the color. She's got the beautiful necklace, and who's a professor at U of O from the Interdisciplinary School of Health Sciences, and Mrs. Talia Salzman, her PhD candidate from the School of Human Kinetics, also from Ottawa U. And so our two colleagues here, I turn the floor over to them, and please, I hope you enjoy your session.
Hi, everybody, welcome. I hope everyone can hear me okay. Bonjour tout le monde. Bienvenue. Je suis contente d'avoir tout ce monde-là aujourd'hui. I'm happy that everyone has been able to attend today. I'm very impressed by the numbers. I'm going to start the presentation, so I will share my screen. I'm going to begin, and then I'm going to pass it to Talia, and she will complete our presentation. So I'm just going to jump into our little presentation here. There we go. So you heard the title of our talk. I'm guessing that maybe some of you are concerned about your memory. Perhaps that's why you're here today, or you're concerned about somebody you love. Well, I like to start my presentations with why I am interested in this and why I'm doing research on this. So I have a little slide here. Why do I care? I started my research in cognitive aging, looking at people's thought processes, how they attend to things, actually their brain activity, mainly because of my grandmother, who's pictured here on the left. This is her at the age of 78, going to my cousin's wedding in perfectly good health. And not long after this wedding, my grandmother had a fall and subsequently had some cognitive decline and actually ended her days in a long-term care home in Brockville, Ontario. And I lived all of this and sort of transitions over time with my grandmother. And so I started asking myself, one, how can we prevent the fall? Two, what is the cognitive processes involved in all of this that could have precipitated further cognitive declines? So a lot of my research and the work that I've been doing with Talia looks at motor control from the point of view of mobility or finger motion, and cognition, so the thought processes and attention that people require while they're doing something motor control and while they're thinking of something cognitive. Because in the case of my grandmother, when she fell, she was walking and talking and she tripped over a rug, and that's all it took, and she broke her hip, and then the declines followed the fall, really. My dad is in the same situation. So I go to Montreal every 3 weeks now because my dad is diagnosed with moderate to severe dementia, and he has mobility issues as well. And my mom, who's 80, is the primary caregiver. So I'm doing this research for him as well. And guess what? I'm pretty selfish. I'm doing this for me as well. So the reality is we're all aging. Things change with age, but maybe there are some factors that hopefully, I'm going to say lots of people are doing this, but Talia and I are trying to identify earlier some kind of changes that might identify somebody at risk, and maybe we can intervene earlier and change the trajectory so that it isn't one with severe cognitive decline. So in terms of our talk today, we're going to talk about first what normal aging is, and then talk a little bit about cognitive impairment. So on this graphic, you can see cognitive function on the y-axis, and then on the x-axis, you can see time. And you'll see when I discuss normal aging means that things change. It doesn't mean that you're going to stay the same as you were when you were 20. Everyone knows this that's around the table. There are some muscle changes, there are potentially vision changes, hearing, and in our case, we're looking at brain changes. But some of this is all a part of normal aging. There are changes with age. Talia is going to talk to you about this acronym SCD, which stands for subjective cognitive decline. And this is something that's a new target that we're looking at for prevention of cognitive decline, where people feel like their memory or their cognition has changed, but there is no objective tests saying that the person has any memory impairment. So this is a new target that we're looking at. And Talia, I'm going to let Talia talk about it, but it is possible, even though the person identifies as having subjective cognitive decline, that really this person is following the normal processes of aging. In addition, if we start talking about cognitive impairment, I'll be telling you a little bit about mild cognitive impairment, or MCI. This is considered a potentially precursor stage to Alzheimer's disease or other types of dementia. In this stage, the changes in memory or the changes in cognition are actually greater than what would be normally expected for your age. So it's sort of a little bit of a red flag. It doesn't mean that there are pathological processes in place, but it's a bit of a red flag that this is greater than what would be expected for your age. In some cases, people who have been identified as having mild cognitive impairment, a percentage of those individuals do progress to a type of dementia. But it is, again, a stage at which, if you identify someone at this stage, you can change the trajectory over time and reduce potentially the declines that somebody faces. And lastly, there's dementia. There are many types of dementia. The most common is Alzheimer's disease, with 60 to 70% of the cases around the world being Alzheimer's disease type dementia. This is a pathological process, and there are typically 3 stages where it is mild, moderate, and severe. I'll give you a few more details on each of these things, but that's sort of the framework of our talk today. I'm going to cover normal aging, mild cognitive impairment, and dementia, and then Talia will complete the talk talking about our new target of subjective cognitive decline. So in terms of what is normal, and I say that with quotes, as I've already mentioned, everyone has gradual changes. Musculature change. In fact, I usually say this to students who are in human kinetics. At the age of 25, muscle loss begins, and you can be a triathlete, you can be whatever, you can train every day of your life, you will still have some muscle loss. This is a normal process of aging. Despite that, everyone changes across age, but not necessarily at the same rate. So there are some people who may age more swiftly, a little more slowly. There are some people who may have hearing loss, other people that have some vision changes, other people that have memory changes. It's not the same for everybody. So the changes are highly variable in every individual. When it comes to memory, in all honesty, and maybe this is the same around the table in the room, almost 40% of adults age 65 or older will identify as having some kind of memory loss. Now, if they go see their doctor and get tested and there is no medical reason for this memory loss, it's considered age-associated memory impairment, which basically means as you age, there may be some changes in your memory, but it's not significantly interfering with you doing your everyday activities. Now, there are things that affect normal aging. You may have read in the news, there are some genetic factors that may increase your risk of cognitive decline and dementia. ApoE4 is one of the genetic factors that's been identified as being a risk factor for the development of dementia and cognitive decline. However, in saying that, I will tell you that only 25% of the population actually present as having one of these genetic factors. And even if you have this factor, it is a risk factor. It does not mean that you will necessarily develop dementia. There's also individual differences in the susceptibility to age-related brain changes. So structure, it sounds funny, but bigger brains are better. Having a larger brain means that you can withstand more infarct to the brain and more damage. And everybody has different sized brains. So this can be a factor in terms of normal aging. And then there's the side of coping with changes in the brain. So in reality, the brain is an amazing feature of our body in that it can reorganize and compensate in some cases for different areas of the brain that have been impacted. So we see this, for example, in stroke, where a certain area of the brain has been impacted, and then neural networks surrounding that area will actually start to try and pick up and compensate for the loss in a certain area. I'll bring your attention to the picture on the screen. So this is a PET scan where they're looking, it's an imaging technique where they're looking at the metabolism in the brain of individuals that are older adults that are normal, have mild cognitive impairment or Alzheimer's disease. And when it's really lit up in red, that means there is a lot of activity happening in the brain. The front of the brain is here. So this would be the prefrontal cortex, and the back of the brain is here. And these are the vesicles in the brain here. And what you can see by looking at normal aging versus mild cognitive impairment versus Alzheimer's disease is that there's a lot more activity in a normally functioning older adult brain. When somebody is diagnosed with mild cognitive impairment, there you see that there is less heat, less red on the map, and greater loss of structure in the vesicle area. And then as you see here with Alzheimer's disease, again, a reduction in brain activity and a much larger loss of brain area, in the center here. And that's the center in particular. That's why Alzheimer's disease is most commonly associated with memory losses, because some of the areas that are affected initially are the hippocampus, which is very important for maintaining memories. A lot of people ask me when I give these talks, is there something I can do? Are there things that I can change? There's a relatively recent paper from Lancet, from the United Kingdom, that has tracked individuals across their lifespan and really identified modifiable risk factors for cognitive decline and dementia, and has pointed out that actually 35% of dementia risk, which is a huge percentage, is related to potentially modifiable risk factors. So if you look at my little graphic here on the right of the screen, one of the early life factors is education. So you increase your relative risk of dementia if you have a lower education level. In midlife, hypertension, obesity, and hearing loss are factors that contribute to increasing your risk of dementia. And in later life, smoking, depression, physical inactivity, low social contact, and diabetes all contribute to increased risk of dementia. Many of these, and I know some of these will depend on circumstance, but some of these are modifiable. So changing your nutrition. In terms of hearing loss, I found this interesting, actually, if you read the Livingston article, that they demonstrated that people who chose to get a hearing aid actually reduced their risk of dementia compared to those who chose not to get a hearing aid. And a large percentage of the dementia risk is related to hearing loss. So some of these things can be worked on: nutrition, exercise, making sure that you get out and you make social contacts. These can all help reduce your risk of cognitive declines. Just to be more specific about what is cognitive impairment, I've mentioned this already. Mild cognitive impairment is typically associated with memory loss and some other symptoms. But again, at this point, it's not thought that there's any pathology, that there's actual major infarct in the brain, but it doesn't interfere with your daily routines. So you can be diagnosed with mild cognitive impairment, and nobody who meets you would know that you have mild cognitive impairment. It's so mild that it really doesn't interfere with your daily functioning. Dementia, the Alzheimer's Disease Society of Canada makes the distinction that this is not normal aging. So this is truly, when you have a diagnosis of dementia, this is when a pathological process is definitely in place. This pathological process affects the brain. It reduces the structure of your brain, so you lose a lot of your brain, your cortex, and it does cause memory loss and other symptoms. It can change mood. It can make you forget common places, forget numbers, how to work with numbers and words, and it does affect your daily routines. So this is definitely different from normal aging, mild cognitive impairment, and even subjective cognitive decline, which I will pass over to Talia to discuss at this point.
Perfect. So in the next few slides, I'll be talking about subjective cognitive decline, some of the research that I'm doing right now as part of my PhD. And at the end, we have some information if anyone is interested in this topic and would like to participate in our ongoing projects. So I'll first just start by discussing what subjective cognitive decline is. We refer to it as SCD. So this is a self-reported decline in cognition. So it has to be persistent, usually over the past year. So this could be from anyone saying, I'm worried about my memory, I have trouble finding words when I speak. But when they go see a doctor about this, they perform normally on the cognitive assessments that they're given. So in other words, it's not detecting what their primary complaint is about. So if we look at this timeline here, I don't think you could see my mouse, but yeah. So if you look at this timeline, subjective cognitive decline as of right now is probably found within the normal range of aging compared to objective measurements, which Sarah talked about a little bit, mild cognitive impairment and dementia. So the question becomes, how do you measure something that's subjective if everyone has different perceptions of what that is? So what we are planning to do and like to do in our lab is to look at brain activity. So as Sarah discussed a little bit, there can be changes that are happening in the brain that actually allow you to compensate for the behaviors you see. So let's say how you're performing on these standardized tests is because something is happening in your brain in order to allow you to perform at that level. So what we want to do is look directly at brain activity and see what's happening in people's brains who are reporting subjective cognitive declines. We can go to the next slide. So the rationale for this and why it's important is that the current tests that are being used to detect cognitive decline aren't sensitive enough to detect the earliest stages or subtle changes in cognitive decline that are happening years before a diagnosis of mild cognitive impairment or dementia. So it's important to identify these people because early detection can increase treatment options. This can be a variety of things. Sometimes it could be an intervention, maybe cognitive training, maybe it could be physical activity in order to reverse or delay cognitive decline. But it can also allow older adults to age in place and maintain their functional independence for as long as possible, which is super important. And lastly, for safety reasons, there's a lot of research on the association between safe cognitive decline and falling. So if we can identify as early as possible people who are experiencing cognitive decline in order to ensure their safety. And in the grander scheme of things, this can help identify other people who are at a risk of future cognitive decline, for example, mild cognitive impairment or dementia. So one example is Alzheimer's. So subjective cognitive decline is an important emerging target. There's obviously a growing aging population with a lot of people reporting cognitive decline. Between 50 to 80% of people take these standardized tests, perform normally, but still have cognitive complaints. But the question becomes how many are among the worried well and how many are actually at risk of future declines. So as Sarah mentioned before, an important brain region to us is the prefrontal cortex. And this is associated with executive functions and it controls executive functions. So executive functions are involved in goal-directed behaviors. So this could be planning, inhibiting. Inhibiting can refer to, for example, if there's a lot of, if you're trying to complete a task and there's different sources of information, just blocking out what's irrelevant and focusing on what's important for the task at hand. And also coordination. So since it's associated with the prefrontal cortex, we also know that it's one of the first brain regions that is most susceptible to deterioration with age. So ideally what we want to do is try to enhance that brain region using non-pharmacological interventions. So what we do know in subjective cognitive decline is that there's actually increased brain activity when people do executive function tasks, for example, working memory. So go to the next slide. So the way we test executive functions is using something called the dual task paradigm. So if you can imagine, your attention or processing capacity has a limit. So the more tasks you do at the same time, the more your attention has to be divided between each task. So specifically the dual task, what we're looking at is 2 tasks at the same time. And we're looking at how you're able to divide your attention between both tasks. And what you could see in the figure on the right here, when you do one task on its own, for example, if you're just walking, you would think that someone's performance or their walk speed would be highest compared to, let's say, when they're walking and talking. And that change in performance, so let's say we're looking at the speed that they walk at, the decrease in speed is what we call a dual task cost. So what we're doing, we're looking at costs both in the brain, so how your brain activity changes, and also different measures of performance, so walk speed. And we also have a second thing that we do with finger tapping. So in our case, what we're doing is we use a motor task and a cognitive task. And yeah, that's the dual task. So in terms of what we're measuring, I spoke about it a little bit in terms of performance. This is what we already know from the literature, is that when we compare subjective cognitive decline to a control group, there's the subjective cognitive decline group has slower response times when they're walking. So for example, if we ask someone to solve a simple math problem when they're walking, they respond with the answer to the math problem slower while they're walking compared if they were just standing and responding to the math question. But then it's also interesting because if we look at a different type of performance, for example, balance, people with subjective cognitive decline demonstrated better balance on a foam surface. So it's less stable than, for example, standing on a firm surface. So if you can imagine what these lines are showing, the blue and red, so it's tracing your balance as you move while you're standing. So you could see the one on the right is a little more sporadic in red, which would indicate worse balance. Then if we compare SCD to further stages of cognitive decline compared to the previous example, which was a control. So if we compare it to mild cognitive impairment and dementia, we can see that using a finger tapping task. So essentially, if you sit at a computer and are pressing a sequence of numbers, like with different fingers, we find that people with subjective cognitive decline have more, or with later stages decline, have more variability in their finger taps. So the rate at which they're tapping, and also they have a decreased number of taps overall compared to an SCD group. So last year, we actually sent out a survey to get some preliminary data on older adults' perceptions of their dual task abilities, and also about their memory and some other cognitive domains. So first, I'll just talk about the figure on the left. So this is about dual tasking abilities. So we sent out a survey and it asks a bunch of different questions about how you perceive your ability to do two tasks at the same time. Some of the questions were walking while having a conversation with someone, or for example, driving and listening to music. And then we asked people to rate their abilities as no change compared to 10 years ago, if they find it easier now or harder. And so what you can see on the scale here, so the y-axis, yeah, there. So the higher the number, the more difficult it is for them. So you could see that the subjective cognitive decline group, which is the red bar, they found it more difficult than people who did not have cognitive complaints, or the subjective cognitive decline group. And then in the figure on the right, this was a survey about memory. So we asked questions, for example, do you remember where you placed an object that you had put down, or remembering appointments? So questions like that. And the participants had to rate whether they had from better or no change to they find it consistently much worse, their memory. So again, the scale on the y-axis, the higher the number, the worse they find their memory. So the red bar, you could see that the SCD group reported that they had worse memory than the non-SCD group. So now what we'd like to add on to that survey, that we know that there are differences in dual task abilities or their executive functions, and that there are differences in their memory, we want to add a neuroimaging component to this to see what's happening in the brain as they do tasks that involve two things at the same time. So the imaging device we used is called functional near infrared spectroscopy. Functional is because we're measuring the function of the brain, metabolism, compared to, for example, the structure of the brain. So we're actually measuring blood flow. Near infrared, it's because we used infrared light. So if you can imagine, visible light is what it's the regular light you see with your eyes, but near infrared would be closer to a red color. And spectroscopy is referring to the study of absorption and like reflection of light. So in that figure on the top right, you can imagine if you had a flashlight and we're shining it through your skin, some of it goes through where some of it gets reflected. So what we're measuring is how much of the light gets reflected back. So what's actually being reflected, as I said, we're measuring cerebral blood flow. So when you're doing a task, there's increased blood flow that goes to the brain region that is being used in that task. So we assume that the more the task is, let's say, difficult, the more brain blood flow goes to that region of the brain, and we're measuring the hemoglobin. So it's a protein that carries oxygen in red blood cells. So we're measuring how much extra oxygen is going towards that region of the brain. So we're specifically looking at the prefrontal cortex. So you can see a picture of our device specifically on the right there. So it's essentially a headband that has 8 sources of light and 2 detectors to measure changes in brain activity. And then the output to us looks like what you see in this figure with the red, blue, and green lines. So the red line is the amount of oxygen going to your brain. The blue line is the oxygen going away from your brain. And then the green line is basically an average, so the total of both. So this technique is very convenient because it's portable. So if you can imagine, you can wear it while you're running, doing exercise. We'd like to do walking, sitting. So it's very convenient in that way, but it's also good because it tolerates different sources of motion from walking, head movements, etc. As you can imagine, if any of you guys know what an MRI scanner looks like, so it's pretty large, you're lying down, so it kind of limits the type of things you can do. So this technique allows us to measure brain activity during multitasking and during walking as well. So if this topic is interesting to you, if you're interested in these sorts of findings, then we're actually for my PhD, and we have some 4th-year undergraduate students that are helping with this project because we want to examine differences in brain activity between older adults who subjectively report cognitive declines compared to others that don't. We're specifically looking for people who are over the age of 60 and who are right-handed, and that's because people with right-handed and left-handed have different or opposite brain activities, so we want to keep it consistent amongst everyone. And we also are looking for people who have self-reported good health, so this would be no neurological diseases, for example, a history of strokes or multiple sclerosis, and also people who don't have hearing or vision impairments, because that can also affect brain activity. And lastly, we're looking for people who can speak English, and this is because some of our questionnaires that we give are unfortunately only validated in English. And just in the picture on the right there, that's the study I did for my master's, and it's just a picture of someone walking along in our lab with their neuroimaging device and all the other equipment we give them. So what would be involved? It would be one session at the University of Ottawa. It lasts between approximately an hour and a half to 2 hours. And then we'd ask you to complete some dual tasks. So the first one would be walking, and we have a second one, which is finger tapping. And then we ask you to do either one of those at the same time as a memory task. So that's where you could see the dual task comes in. So walking with memory or doing a finger tapping task. So pressing buttons on a keyboard with the memory task. And then at the same time, while you're doing that, we'll measure your brain activity. So this is non-invasively. As I said, it's just with light. And then you can press the slide in. Yeah, so after that, you'll complete a few short questionnaires and neuropsychological tests. So these are paper and pencil tests. And then we'll also compensate you $30 for volunteering your time. So we're located at the University of Ottawa Lees campus. So it's about a 5-minute drive from the main campus of the University of Ottawa. It's located at 200 Lees Avenue. We have reserved parking on site if you want to drive in. We also have can reimburse for public transportation. So we're right across the street from the Lees LRT stop / bus stop at Lees. And then we also have the University of Ottawa has a few COVID protocols in place. So everyone has to wear a mask while indoors. So we'll be wearing a mask as well as face shields. We also sanitize all our equipment and the lab space. And also there's a policy at the university that everyone has to be vaccinated. So we'll all be vaccinated. So we have also some other ongoing studies, and Sarah, feel free to jump in here if you want to explain them. But so the first one, these are both virtual. The first one is based on an app that our lab developed in collaboration with some engineers. It's called the Safer Steps app. And basically it's measuring, you would download the app on your phone or tablet, and then you can assess how safe your stairs are compared to building regulations, safety regulations, because we know that there's risk of falling on stairs in older adults. So essentially what you would need to do, you would be in a focus group, so with approximately 4 other people, and then you would just report on your experiences using the app because we'd like your feedback on that. So that would be over Zoom, and it would take between 45 to 60 minutes. And then we also have another one, which is talking about caregivers and their experiences caregiving for someone during COVID-19. So this would be a one-on-one interview. This would be over the phone. And you would just describe your experiences being a caregiver, and also if you've contemplated transitioning the person you're caring for to long-term care, what that process is like, and the resources that you've had or the experiences you're having. So again, that's over the phone, and that would be between 45 to 60 minutes. So if there's any other questions, feel free to email us. The first email there is our lab email. So we're constantly monitoring that. If you have any questions about any of these studies or would just like more information, feel free to email that address. And then the second one is Dr. Fraser's email, and we're also reachable by phone with the phone number at the bottom. So we're happy to answer any of your questions about the studies.